But now we have to see what things can affect this. Now let's start there. Let's say that you're actually going to be, let's say we come over here to the blood first. Let's start with these chemoreceptors, because these are usually the harder ones to do. We'll start with the central, then we'll do the peripheral. Let's start off first, and let's say that you have high partial pressure of CO2 within the blood. Okay? You have a high partial pressure of CO2 in the blood. So what does that mean? That you have more CO2 in the blood. If there is excessively large amounts of CO2 in the blood, what does that mean? Okay, well, you know, let's say right here, this is specifically let's say that this is actually the cerebrospinal fluid. Okay, so this is the cerebrospinal fluid, and this is where your neurons are. This is where the central chemoreceptors are. So if I were to actually draw this here, let's say right over here is my central chemoreceptors. Central chemoreceptors. And I want to stimulate them. Well, I have to cross between the blood and the brain barrier. So in order for me to do that, I have to bring the CO2 in, because you know protons can't get in, right? Because you guys know that protons don't have the ability to get in because they're charged. So CO2 will actually come into the cerebrospinal fluid. It'll cross the blood-brain barrier. So again, what is this structure here called? This right here. This is actually the blood-brain barrier. When CO2 crosses the blood-brain barrier and goes into the cerebrospinal fluid, it actually will combine with water. When it combines with water, it'll actually fuse with the actual water to form carbonic acid, H2CO3. When H2CO3, which is carbonic acid, is formed, it's very unstable, and what it'll do is it'll actually dissociate into two different chemicals. One of the chemicals is actually going to be protons, and the other one is going to be bicarbonate. Why am I telling you this? Because whenever there is an increase in the amount of CO2, that increases the amount of CO2 here, that increases the amount of carbonic acid, and that increases the amount of protons. If there's more protons, that's actually going to stimulate the central chemoreceptors. Then what are the central chemoreceptors going to do? The central chemoreceptors are going to respond to that stimulus. And then they're going to send signals. So now let's pretend that this central chemoreceptor is this one here. What it's going to do is it's going to stimulate the actual, let's do this in a bright color. Let's do this in a pink color here. This central chemoreceptors are going to stimulate the DRG. Okay, they're going to stimulate the DRG. You know what else the central chemoreceptors are going to do? They can also stimulate the pneumotaxic center. And then that can send signals down to the DRG. We'll show that very small here. It also can affect the pneumotaxic center. And then the pneumotaxic center can send these signals down to the DRG also, as well as to the VRG. We're going to specifically stick here with the DRG in this case. But it can send signals to the VRG, right? So if the central chemoreceptors are stimulated due to these high protons, what does this mean whenever you have high protons? What does that mean for the pH? It means the pH is decreasing. So you have a decrease in the pH. When these are stimulated, the DRG can send down inspiratory signals. But guess what else the DRG will do? The DRG is so kind. He'll tell some of the inspiratory neurons within the VRG, right? So he'll come over here and he'll tell some of the inspiratory neurons within the VRG. And he'll say, hey, VRG, send some inspiratory signals down. And that's what the actual VRG will do. So it'll also send inspiratory signals down. If both of these guys are sending inspiratory signals down, what's going to happen to the actual frequency of the impulses moving along the actual intercostal nerve? Okay, well, these are going to increase even more. If these increase even more, what does that mean for the actual phrenic nerve and the intercostal nerve? That means that these are going to stimulate the diaphragm and the external intercostals even more heavily. What's that going to do? That's going to, so again, what should I say here? I should say the frequency of action potentials. So what happens to the frequency here? So the frequency is actually going to increase. So the frequency of the action potentials is going to increase. And so if that's the case, it's going to cause more frequent contractions of the diaphragm and the external intercostals. If they contract more frequently, what is that going to do? That's going to increase ventilation. So what is that called whenever you're doing this? The overall result of these two effects here is to hyperventilate, right? To increase the actual ventilation process. Okay, now when I say hyperventilation, I obviously need to be careful because it depends upon what the actual initial stimulus is. because it could be hyperventilation or it could be hyperpnea. We don't know exactly why the CO2 levels are rising. So I'm not even going to put hyperventilation. I'm just going to specifically say there's an increase in the ventilation. So let me just put that. There's an increase in the actual, I'll put this, increase in the respiration rate and the depth, right? Because now, let's think about this for a second. If you have a lot of CO2, so let's come over to this corner part of the diagram. Let's come over here and let's say that you have, in the blood, high amounts of CO2. If you have this increase in respiration rate and respiration depth, so I increase the ventilation process, in other words, because that's all I'm saying. When I say increase respiration depth, I'm just basically saying increase my alveolar ventilation. That's all I'm saying, really, alveolar ventilation. So now I'm going to have more air coming in, and I'm going to try to cause more air to go out. If I increase that ventilation process, what does that mean? There's going to be more gas exchange. More CO2 is going to be coming into the alveoli, and then what? Leaving out into the atmosphere. I'm going to try to breathe off more CO2. Then what's going to happen? More oxygen is actually going to funnel into the blood. And then what's that going to do? If I exhale a lot of CO2, what's going to happen to the CO2 levels in the blood? It's going to go down. As the CO2 levels go down, what happens? Let's find out. Let's go back over to this diagram. If the CO2 levels decrease, oh, okay, well, if the CO2 levels, and let's just say this is appropriate decrease, let's not say it's decreasing significantly, like really, really bad. Let's say it's coming back down to normal. So it's going to decrease the CO2. Okay, that's cool. It's going to decrease the amount of carbonic acid. It's going to decrease the protons. And if it does this and brings it back to normal, doesn't that take the stimulus away from the central chemoreceptors? Yes. And if that's the case, then the DRG will not be sending impulses down, as well as the VRG, and the respiration will start to slow down a little bit, right? And come back to normal. So that's the whole purpose. So whenever the CO2 levels are high, it's going to increase the respiration rate and the respiration depth. What happens if our CO2 levels are significantly low? So now let's represent this with a different color to show that we're not talking about normal situations. Let's say that we're talking about very, very low. What was that called whenever you have really high partial pressure of CO2? This is called hypercapnia. Now we're going to talk about what happens whenever you have a low partial pressure of CO2. Low partial pressure of CO2 is referred to as hypoxemia. Okay, so now let's take the opposite effect. Let's show this one, and let's do it in black since we already wrote that in black. If there's a decrease in the CO2, that means there'll be a decrease in this CO2. If there's a decrease in that means there's a decreased formation of carbonic acid, which means that there's a significant decrease in protons. If the protons are so low, they're not going to be providing signals to the central chemoreceptors. If the actual central chemoreceptors are not going to be as frequently stimulated, so let's actually put inhibited in this case. Even though we know they're not inhibited, we just know that they're not going to be receiving a lot of stimulus. If they're not receiving a lot of stimulus due to these low proton levels, are they going to stimulate the DRG? No, so we can think about them not stimulating the DRG. So let's think about this for a second, and we'll write it the opposite way. We'll say it's inhibiting the DRG, okay? But again, just remember that there's very little signals going to the DRG. Then we can also say that there's very little signals going to the pneumotaxic center, right? So very little signals going to the pneumotaxic center. And this will also affect the DRG, and it won't be sending as much impulses to the DRG. If this is not sending a lot of impulses to the DRG, then the DRG will send very little impulses to the VRG. And you should be getting the point already now. That now, if the VRG and the DRG are inhibited, then what's going to happen? That means that the impulses that are actually going to be being sent down to the actual phrenic nerve, this is going to decrease. These impulses are going to decrease. If these impulses are decreasing, then what's going to happen? The frequency is going to decrease. If the frequency decreases, what's going to happen to the rate at which these muscles are contracting? Well, it's going to be a slower rate of contraction, right? So in other words, when the diaphragm contracts and when the external intercostals contract, it's not going to be happening at a very high rate. So what's going to happen to the respiration rate and the respiration depth? That will decrease. And if the respiration rate and the respiration depth decrease, what's going to happen? Let's come back over here. Okay, well, if there's really, really low CO2 levels, okay, and let's get rid of this right here for a second. really, really low CO2 levels, and we're decreasing the actual respiration rate and the respiration depth. So in other words, we're hypoventilating. Let's say that we're hypoventilating, and our ventilation is decreasing. That means it's going to be very slow, very, very shallow breathing. Whereas before, when it was high CO2, we were actually, there was an increase in the respiration rate and the respiration depth. Now there's going to be a decrease, so it's going to be very slow and shallow breathing. That means that very little CO2 is actually going to be moving across this actual respiratory membrane. If very little CO2 is moving across this membrane, what happens to the CO2 that's going to be exhaled? The CO2 that's going to be exhaled is actually going to be a lot lower. So what does that mean then? So if there's actually this consistently low output of CO2, that means that some of this CO2 in the blood is going to start slowly building up because we're not getting rid of a lot of it. So we're not ridding it out of the blood and putting it into the alveoli and then exhaling it. Very little of it is actually being exhaled. So it's actually being retained in the blood due to this slow, shallow breathing. If that's the case, then what's going to happen? Let's come back over here. If that happens, then what will happen to the CO2 levels in the blood eventually, right? It'll slowly start rising. If it slowly starts rising, this will go up. As this goes up, this will go up. And as that goes up, this will go up. And then what will happen? This will no longer be inhibited, and it'll start being stimulated, right? To allow for normal respirations to begin again. And then if he's stimulated, then this guy will be stimulated, and you're going to get the point here. This is stimulated, and then this would be stimulated, and then this guy would send stimulation, and then the frequency would go back to normal, and then we'd have normal respiration rate and normal respiration depth, assuming that we're doing this normally, right? But again, the whole concept is that what is these whole inspiratory centers trying to do? They're trying to control the pH in response to the changing, fluctuating CO2 levels. Because you know, the partial pressure of CO2 is the most powerful respiratory stimulant, unless oxygen levels drop below 60, and then it becomes the most powerful. Okay, so understanding that is critical. Okay, so that's that for the central chemoreceptors. Okay? Now what we're going to do is we're going to hop over here and we're going to see how the peripheral chemoreceptors are playing a role in this. Very similar, but they're responding to pH in a different way. Okay, so those were the central chemoreceptors. You see these little blue dudes here? They're right around. Well, first off, let's look at our anatomy here real quick. So this right here is actually going to be the left atrium. This is the left ventricle. Then coming up out of here is actually going to be your aorta. Then off of the aorta, you have your first artery here. You have your brachiocephalic artery. So I'll put here BA. Then off of the brachiocephalic artery, it splits. It goes into the right subclavian artery. And then it goes into the common carotid artery. And this would be the right one. This is the left common carotid artery. And then this is the left subclavian artery. And then again, it'll go down to the descending aorta. Then the right common carotid artery will come up here and split. Well, at that splitting point, when it goes into, let's say that this is the external carotid artery, internal carotid artery, internal carotid artery, this one will be right, this one will be right, this will be left, and this will be the left external carotid artery. That point where they're bifurcating, there's these specialized chemoreceptors. And they're in these actual body formation, right? So these peripheral chemoreceptors are right here. They're located right here on this point here. What is this point right here called? This point here is actually called the carotid bodies. Okay? And that's where some of the peripheral chemoreceptors are. The other ones are going to be right here in the aorta. This is actually called the aortic bodies. So these are called the aortic bodies. Now, these are, again, your aortic bodies and your carotid bodies are actually a component of what's called your peripheral chemoreceptors. Okay. Now, the peripheral chemoreceptors are special because they respond to 3 different stimuli. Let me write those stimuli down. They can respond to partial pressure of CO2. So they can respond to a partial pressure of CO2. They can respond to the partial pressure of oxygen. And they can respond to the pH. But we're going to go into a little bit more detail on this. Okay. So now, the peripheral chemoreceptors are special because we said that they can respond to CO2 changes, they can respond to oxygen changes, and they can respond to pH. out of all of these, the most significant one that we need to remember out of all of these is going to be the partial pressure of oxygen. So whenever the partial pressure of oxygen drops below 60 millimeters of mercury, it is the most powerful respiratory stimulus out of all of the other certain types of chemicals. It's greater than the CO2 stimulus and greater than the pH stimulus. Now, generally, oxygen is usually normal, right? In certain situations, the partial pressure of oxygen is usually normal, so we're assuming that it's greater than 60, and so CO2 is the most powerful stimulus, and then pH is also contributing to this. Now, CO2 is affecting the central chemoreceptors. 70% of it is affecting the central chemoreceptors. 30% of it is only really affecting the peripheral chemoreceptors. But again, oxygen is the most powerful stimulus. And here's another thing for the pH. The pH is not due to the changes in CO2. It's due to the changes in metabolic acids. So like what? Like ketone bodies. You know, these are produced whenever you have prolonged starvation or in type 1 diabetes mellitus that's not properly controlled. Or lactic acid. Or other different types of metabolic acids. We can keep going, right? The whole point is metabolic acids are the main influence of this arterial pH, not the fluctuations in CO2. Okay, so again, peripheral chemoreceptors, they're responding to the partial pressure of CO2. About 30% of that actual CO2 is going to be, specifically stimulating the chemoreceptors, right? And then if the partial pressure of oxygen is below 60 millimeters of mercury, it becomes the most powerful respiratory stimulus. And the last thing is that these actual peripheral chemoreceptors are specifically responding to changes in pH due to metabolic acids like lactic acid, ketone bodies, and stuff like that. Okay? Okay, so now let's see the mechanism of how this is working. So let's see the mechanism of how this is working. So here I'm actually zooming in on the actual carotid body or the actual aortic body. Now, in these carotid bodies, aortic bodies, you have two types of cells. This is actually called the type 1 cell or the type 2 cell. Now, technically they call it the type 2 cell. They call it, it's like the sustanacular cells, not the sustanacular cells in the testes, so don't get that confused. So sustanacular cells, because it's basically a supporting cell. And this type 1 cell is actually called your glomus cells. Okay, anyway. In the glomus cells, they actually consist of these packages, these vesicles of neurotransmitters, specifically that of dopamine. So that's consisting of these actual dopamine-containing vesicles. Now, the whole purpose of this is we either want to cause the release of dopamine or we actually don't want to release the dopamine. So let's see how we actually do this. Now, in this actual glomus cell, you have specialized potassium channels. So you have these specialized potassium channels. Now, these potassium channels have three different stimuli, okay? So here's our potassium ion. We're going to determine whether the potassium ion is going out or in. Generally, this potassium channel is sensitive to oxygen, protons, so oxygen and protons. Now, let's say that your oxygen levels are decreasing. They decrease below what? They decrease below 60 millimeters of mercury. They become the main respiratory stimulus. What are they going to do? When oxygen levels are low, hypoxia, it inhibits these actual channels. It inhibits these channels. Now, let's say that the actual proton levels are going up. What does that mean when the proton levels go up? That means that the actual pH is going down, right? In that situation, that is also inhibiting these actual potassium channels, closing the potassium channel. And the last one, if CO2 is rising, CO2 will come into the cell and combine with water. When it combines with the water, what it will do? CO2 and water will fuse together in the presence of an enzyme, right, called carbonic anhydrase. That enzyme would have been over there with the central chemoreceptor area. What it's doing is it's fusing them together and forming carbonic acid, and then carbonic acid is dissociating into protons and bicarb. Those protons are inhibiting this potassium channel. So these protons here are coming from metabolic acids. But these protons here are coming from the CO2, right? So how does the CO2 actually inhibit this channel? It's directly through these actual protons. So CO2 will come in, get converted into carbonic acid, break down into protons and bicarb, and the protons will inhibit these potassium channels. But these protons, these single protons, are actually coming from metabolic acids like lactic acid and ketone bodies like acetoacetate and beta-hydroxybutyrate, things like that. And whenever the oxygen levels are less than 60 millimeters of mercury, that'll also inhibit this channel. If these channels are inhibited, can potassium leave the cell? No. So potassium can't leave the cell. If the potassium levels accumulate in the cell, what happens to the actual inside of the cell? The inside of the cell becomes more positive. If the inside of the cell starts becoming more electropositive, it leads to an activation of these specialized calcium channels. And when these calcium channels open, they are the ones who are going to make the difference. Look at these suckers here. So look, here's the calcium channel. And we'll put another calcium channel over here. These calcium channels are stimulated due to these potassium ions increasing and increasing the actual membrane potential. When the calcium ions rush into these actual glomus cells, they trigger these actual vesicles to fuse. And when the vesicles start fusing with this actual glomus cell membrane, what happens? Well, you're about to find out. Here we go. Look at this. It fuses, and what starts getting released? The actual dopamine. And whenever the dopamine is released, what happens? Dopamine is going to act on these actual nerve terminals. What are these nerve terminals here? It depends on which body we're talking about. Let's look and see. Oh, it's a beautiful mechanism. I think that's so cool. Okay, that nerve terminal right there, it could be connected with the carotid bodies, or it could be connected with the aortic bodies. If this actual nerve terminal is connected to the carotid bodies, it's specifically going to be cranial nerve 9. There's a 1 before the X. All right. Cranial nerve 9, glossopharyngeal nerve. If it's coming from the aortic bodies, it's cranial nerve 10. And that is the vagus nerve. So again, if these actual carotid bodies are stimulated due to low oxygen, high protons due to metabolic acids, or CO2, which indirectly gets converted into protons and inhibits this potassium channel, causes the inside of the cell to become more positive, activates calcium voltage channels, which come in and cause dopamine to be released, right? Now imagine if all of these things were opposite. It would be the exact opposite thing. So imagine if CO2 levels were actually low. If CO2 levels were low. If oxygen was normal. And if the pH was high. So we did everything whenever CO2 was high, PaO₂ was low, and pH was low, right? And the opposite. If oxygen is high, it's going to stimulate these potassium channels, and potassium will go out and will not cause the calcium to come in. No dopamine will be released. Holy crap. If the actual pH is high, that means that the protons are low, and they won't be inhibiting the potassium channels, and potassium will be able to leak out. If CO2 levels are low, it won't be forming as much protons, and it won't be inhibiting this channel. It'll be stimulating it and allowing for potassium ions to come out, and will prevent the cell from releasing dopamine. So you can imagine if dopamine is being released, what happens to the actual signals? The signals will increase. So if that happens, these signals carried on, cranial nerve 10 and cranial nerve 9 will be increased. If there's very little or no dopamine release, the signals will be not present, or very, very low signals. Okay? Beautiful. So now, let's take those guys and see what they're doing now. Let's bring them up over here. So we're going to say, we'll put them right here. Let's say here's those, right? Here's the other one. And again, what is this coming from? This one is coming from, let's say this one here is cranial nerve 10, which is the vagus. This is cranial nerve 9, which is glossopharyngeal. We know that the cranial nerve 9, which is glossopharyngeal, are responding to the carotid bodies. So acute, right? And then the cranial nerve 10 is responding to the aortic bodies. And again, if there's low pH, due to meaning high protons, oxygen is below 60, and CO2 is elevated, it can actually stimulate these and send action potentials down these suckers here. If the pH was opposite, if the pH was high, the CO2 was low, and the oxygen was normal, the action potentials would not be being sent here. Now, look what they do. They take these signals in to the DRG. It's amazing. If they stimulate the DRG, what's it going to do? The DRG is going to stimulate the VRG. If the VRG and the DRG are stimulated, what's going to happen to the impulses going down to these descending pathways? It's going to increase. If the pathway is going downwards increase, what happens to the frequency of the action potentials coming out here to the phrenic nerve and the intercostal nerves? That's going to increase. If that increases, what is that going to do then? It's going to cause more frequent contractions of the diaphragm and the external intercostals. What's that going to do? Increase the respiration rate and the respiration depth. In other words, you're increasing the ventilation. If you increase the ventilation process, what's that going to try to do? Let's come over here, where your CO2 levels are high. If you increase the ventilation process, you're going to move more CO2 across this membrane. If you have more CO2 moving across this membrane and more oxygen across this membrane, right? So let's say that the stimulus was low oxygen. It's going to cause increased ventilation to get more oxygen into the blood. If the stimulus was low CO2, it's going to stimulate the if the stimulus was high CO2, it would actually stimulate this, increase ventilation across the membrane, which will cause more CO2 to get exhaled. So more CO2 would get lost, and the CO2 levels in the blood would decrease, and the proton concentration would decrease. But there's another problem. What about the metabolic acids, like ketone bodies? You can't exhale ketone bodies. You can exhale what's called acetone, which is a breakdown product of them, but you can't exhale them. And so in situations in which you have this change in the pH, like a decrease in the pH or an increase in the pH, but for right now we're talking about decrease. If the pH is decreasing due to metabolic acids like lactic acid and ketone bodies, you can't breathe these out. If this is due to a situation like metabolic acidosis, how would you treat this? Well, one way is your actual buffers will come into play. Your bicarbonate carbonic acid buffer system will come into play, but it's going to get, sucked up and used up pretty quick, within seconds. Then you're going to try to do this respiration, but it's only going to breathe off CO2. It's not going to take care of the real issue, which is these ketone bodies and lactic acid. You're still going to be stuck, but it might only last for a little bit of minutes. Then, how would you deal with these actual ketone bodies and lactic acid? Your kidneys. So your kidneys would be long-term compensation mechanisms, and they would urinate out the actual ketone bodies, and they would urinate out the protons of the lactic acid. And they would try to bring more bicarb into your bloodstream, okay? So that is the way that your actual body would deal with this. So breathing them off isn't going to help you at all. That's why whenever you see people who have uncontrolled diabetes mellitus, what does their breath smell like? It smells like acetone, because they're trying to breathe off the acid, but all they're doing is actually breathing off CO2. And that's why people who have uncontrolled type 1 diabetes mellitus, they have what's called Kussmaul's breathing, which is very, very abnormal breathing, okay? All right, so that covers the peripheral chemoreceptors and what they're doing, right? And then again, let's apply what happens whenever there was the opposite. So this was being stimulated due to what? Let's actually do this here. This was being stimulated, we'll put here stimulated by what? Low partial pressure of oxygen. Has to be less than 60 millimeter of mercury. It was also being stimulated by high partial pressure of CO2, and it was also being stimulated by low pH, but it was due to metabolic acids. Now let's say the opposite. Let's say that you want to inhibit this one. So now you're trying to do the opposite. You want to inhibit these carotid bodies and the aortic bodies. Then you would want to do what? Do the exact opposite. Have normal partial pressure of oxygen, okay? Decrease the partial pressure of CO2 and increase the pH. And if that happens, let's see what that would do. So if these are inhibiting the actual carotid bodies and the aortic bodies, the impulses that are coming from the aortic bodies and the carotid bodies via the glossopharyngeal nerve, cranial nerve 9, or vagus, cranial nerve 10, is going to be decreasing or almost absent. So what will that do to the DRG? It will inhibit the DRG. If the DRG is inhibited, what is it going to do? It's not going to really send down a lot of action potentials down to the phrenic nerve and intercostal nerve. It's also not going to stimulate the VRG. It might actually slightly inhibit the VRG. So the impulses going down to the actual phrenic nerve and intercostal nerves are actually going to decrease, and then the frequency of the action potentials going to the actual external intercostals and the diaphragm is going to decrease, which is then going to decrease the ventilation, and it's also going to, again, it'll decrease the respiration rate and the respiration depth, which in other words is a decrease in the alveolar ventilation. Holy crap, if you decrease the alveolar ventilation, in other words, what was wrong here? Your CO2 levels were low, If you decrease the actual alveolar ventilation, what does that mean? You're having very slow, shallow breathing. Whereas whenever it was low pH, high CO2, low oxygen, you were having very rapid and very, very deep breathing to bring in more oxygen, exhale more CO2. Well, now it's very slow and shallow. So very, very little CO2 is getting exhaled. And if very little CO2 is getting exhaled and very little oxygen is moving across here, what's going to happen? Okay, well, if there's very little CO2, that means I'm not going to exhale a lot of CO2. So I won't exhale a lot of CO2. So a lot of the CO2 is going to maintain in the bloodstream. Also, if that's the case, then what would that do? Okay, well, if there was a low partial pressure of CO2, we said that that would decrease the carbonic acid formation, decrease the protons, and that would also decrease the actual action here on these actual carotid bodies, aortic bodies. Same thing when there was metabolic alkalosis. If there's metabolic alkalosis due to a decrease in the actual, maybe there's an excessive amount of bicarbonate, you're actually going to want to slow down the respirations, right? So if there's a lot of bicarbonate building up causing metabolic alkalosis, so that's another thing. We said that it was due to metabolic acids like ketone bodies and lactic acid, but you could also change the pH due to other things like, besides these things, we could actually say metabolic bases, right? And if those metabolic bases, they would be things like bicarbonate. So these would be a base. So if there's an excessive amounts of him, that could cause metabolic alkalosis, which actually would do what? Increase the pH. You're not going to want to increase your respiration. You're going to want to decrease your actual respiration so that you accumulate more CO2, accumulate more carbonic acid, and then have that dissociate into more protons, right? Okay. So that's the whole goal there. So we did peripheral chemoreceptors, we did central chemoreceptors, sweet deal. Now let's go ahead and talk about some of these actual receptors over here in lungs.